Welcome to this explainer. Today, we're jumping right into the intense, totally hidden science in the incredibly delicate ecological balance it takes to maintain a healthy aquaculture environment. Specifically, we're zeroing in on the science behind healthy aquatic ponds and how we cultivate white-leg shrimp, also known in the science world as Litopenaeus vanameae. So what does it actually take to keep a million shrimp happy and healthy? Well, instead of picturing your standard, everyday agricultural farm, I want you to imagine an aquaculture pond as a living, breathing patient in an intensive care unit. In this massive ecosystem, scientists and farmers are basically acting as doctors, constantly monitoring the patient's vitals. It is a world of nonstop observation where biology, chemistry, and environmental engineering all have to work in absolute perfect harmony.
All right, let's use this quick roadmap to navigate our explainer today. We are going to cover 1. Preparing the perfect pond, 2. Monitoring the water vitals, 3. Microscopic threats and diseases, and 4. Optimizing nutrition and growth. Let's get into it.
Section 1. Preparing the perfect pond. Preventative care and hygiene. We're talking literal surgical-level hygiene here to break the cycle of pathogens from any previous harvests and prep a totally stress-free habitat. The data shows us that pond preparation honestly dictates the success of the entire cycle. Long before stocking any post-larvae, farmers actually dry out the pond bottom completely to eliminate organic sludge and oxidize harmful toxins. Then, in earthen ponds, they apply lime to stabilize the soil's pH. Once it's clean, water is brought in, heavily filtered to keep out pests, and then strictly sterilized. By locking in stable parameters right from day one, farmers drastically cut down on that initial stocking stress for the juvenile shrimp. Moving on to Section 2, monitoring the water vitals, the heartbeat of the pond. Think of this just like a nurse drawing blood for lab work. Technicians test water samples routinely, round the clock, because even the most minor fluctuations can cascade into catastrophic ecological failures. It's a highly intensive process. The data actually tells us that water quality is the single most important natural indicator for evaluating if a shrimp farm is even feasible. These routine checks, they are literally what prevent sudden, massive mortality rates. We can broadly split these vitals into two camps: physical conditions like temperature and dissolved oxygen versus chemical markers like alkalinity, nitrite, and ammonia. And the optimal ranges here are incredibly strict. Water temperature has to sit right between 27 and 32 degrees Celsius, and dissolved oxygen must be at least 4 parts per million, or ppm. Just to put that into perspective, juvenile shrimp are completely unable to survive if that oxygen drops to 0.65 milligrams per liter. They just won't make it. So the crucial takeaway here is just how insanely sensitive these environments are. The maximum allowable total ammonium nitrogen, or TAN, is less than 0.1 parts per million before it becomes highly toxic to the culture. We are talking about a microscopic fraction. To maintain this delicate balance, absolutely everything, from the pond's water exchange to the paddle aerators churning the water, has to be perfectly, meticulously calibrated. Dissolved oxygen is a highly dynamic vital sign. It hits its lowest point at night, specifically around 10 p.m., and it peaks around 1 p.m. Why? Well, it's all about photosynthesis in the water column during the day. When the sun goes down, all that photosynthetic activity just stops cold. But the respiration of the shrimp, the microorganisms, and the decomposition of organic matter, that all keeps going, constantly draining the oxygen levels in the dark. And this isn't just some dry math on a chalkboard. Regression models from the source studies show that failing to maintain these exact oxygen levels directly stunts the metabolic growth of the entire population. Specifically, those fluctuations in dissolved oxygen levels affect shrimp growth by about 4.4%. It totally alters the aerobic respiration and the oxidation reduction processes happening in the water and deep down in the sediment. Section 3. Microscopic threats and diseases. The invisible battleground. Now, what is truly fascinating and a bit terrifying is how a specific genetic plasmid turns an otherwise standard aquatic bacterium into a highly lethal pathogen. This disease, known as AHPND, affects both black tiger shrimp and the American white-leg shrimp we're focusing on today. The PIR-AB gene actually synthesizes specific insecticidal toxins that are absolutely essential for the disease to take hold and just decimate the ecosystem. If a pond does get infected, these are the severe clinical symptoms our patient is going to display, giving farmers an immediate red alert that they have a crisis on their hands. The disease strikes incredibly fast, causing unusually high mortality within just the first 35 days of culture. The shrimp become moribund, basically sinking straight to the bottom. Their hepatopancreas, which is a vital digestive organ, shrinks, discolors, and turns this pale or whitish color due to a total loss of pigment. Because these bacterial threats are largely invisible to the naked eye right up until the symptoms manifest, routine physical inspections are non-negotiable to visually confirm the health of the population. Technicians have to constantly pull samples and check the physical parameters of the shrimp themselves.
The contrast is night and day, really. A healthy specimen is agile, it's transparent with zero red or black spots, and it's got a full gut. A sick one exhibits clear physiological stress and a total loss of appetite. Healthy shrimp will gather super quickly at feeding trays, so a sudden drop in feed consumption is often your first early warning sign of stress or disease. Sick shrimp also swim abnormally. They'll crowd at the surface or huddle in the corners of the pond instead of using the whole water column like they should. Section 4. Optimizing nutrition and growth, tailored diets, and biofloc. Get this, it's actually super counterintuitive. Studies prove that a 35% crude protein diet actually outperforms a much more expensive 45% protein feed, as long as it's integrated with the right density of bacterial biofloc. You'd probably think more protein automatically equals bigger, healthier shrimp, right? Actually, no. The data clearly demonstrates that 35% crude protein induces the absolute best growth potentials when the biological floc density is optimized in the water. This functions exactly like tailored sports nutrition for athletes. It utilizes the pond's very own microbial environment as a supplemental food source to massively boost efficiency. The biofloc actually improves extracellular enzyme activities, like amylase, which contributes heavily to shrimp growth. It guarantees an adequate supply of essential amino acids without farmers having to waste money overfeeding crude protein. But wait, there's a catch. Feeding too much increases nitrate waste, which instantly triggers physiological stress. This proves that dietary intake and water vitals are intricately, permanently linked. High nitrate concentration kills feed intake, spikes oxygen consumption, and causes mortality. In fact, research shows that nitrate is the parameter with the greatest influence on shrimp growth rates, impacting them by over 75%. If the water gets too acidic and nitrate levels spike, it reduces the retention of nutrient intake and trashes the shrimp's immune system so severely that they will slowly die off. It takes an unbelievable fusion of chemistry, biology, and environmental engineering to maintain this balance. It really transforms our view of what aquaculture truly is, doesn't it? From the daily swings of dissolved oxygen to microscopic battles against lethal plasmids, the science of healthy ponds is a modern marvel of precision. So, what do you think? Does knowing about this intensive, ICU level of scientific monitoring change the way you look at the ecosystems behind the food on our plates?